Hi class, today we're going to be talking about nematodes. So Nematoda is a phylum. Collectively they're known as roundworms or eelworms. This phylum tends to have male and female organisms. The word for that is dioecious instead of hermaphroditic organisms. This phylum also has what's known as sexual dimorphism, meaning that the males and the females tend to look distinct from each other. They have an epidermis that is covered by a thick cuticle, and it must be shed in order for the worm to grow. They have a tubular digestive system, and they have openings at both ends for food to enter and for waste to exit. And their body type is known as pseudocoelomate, which means a tube within a tube. So here's just the general anatomy of a nematode. So here their mouth or their oral cavity contains digestive enzymes to help them begin breaking down any food that they uptake. The pharynx connects directly to a muscleless intestine here, and that forms the main length of the gut, so therefore they don't have a stomach, they just have one long digestive system. And when they're inside of a human, they feed on blood or intestinal chyme, which is just already partially digested food. And here we can see a cross-section of their body, and you can see that pseudocoelomate tube within a tube body style. The way they have their intestine is kind of one hole, and then surrounding that they have certain muscles. And then here is showing the cuticle body wall. So they just have a singular nerve that runs through their body. They only have longitudinal directional muscles. The females possess an oviduct, which is a tube where the egg will pass through when it leaves the ovary. So here's further showing the general anatomy of a nematode. So males have a copulatory bursa and spicules that are involved in grasping the female during mating. You can see here. Males have a cloaca, which is one opening that releases the excretory products and the sperm. And interestingly, the sperm cells of nematodes doesn't look flagellated like how humans or other animals look. Instead, they're pseudopods, and so the sperm cells move around through pseudopod movements. So here you can see overall what the males and the females have similarly is they have their intestinal systems, but then here in the females you can see the ovaries, or instead in the males you have the seminal vesicles. The female has the vulva opening, where males instead have the vas deferens that carry sperm down to the exit here. So here's the development of a nematode. So each larval stage and each different stage is defined as different L's, and then with a different number. So you have L1, L2, L3, L4. So those are the different larval stages. So the larvae must undergo molts in order to increase in size. And so basically the larvae look very similar to the adults, they just grow in size. And starting at L3 is the stage when they'll start to become infectious. So then here you can see it starts with the fertilized egg. Cleavage occurs within the cells within the egg in order to form an embryo, and then they then hatch. They then have to molt and then become the adult. And then here in this image to the right you can see here's the cleavage of the cells within the egg, and then the formation of the embryo, and then images of the different larvae and the adults. And here's just a more detailed transverse cross-section of a nematode. So we have C here, the cuticle, that is a hardened shell, kind of, and they have to shed it in order to molt. Here, their dorsal nerve. Here they have an excretory canal, their hypodermis, just the skin underneath the cuticle. They have their longitudinal muscles. They have a nerve here, which will extend into the main nerve. The pharynx here, which is what leads into their digestive tract. And the way the pharynx has this triradial look to it gives it more efficiency in order to move products down more effectively. And then here the ventral nerve, which is one of the main nerves. Okay, so talking about today's first parasites, we have Encyclostoma, duodenale, and Necator americanus. So both are referred to as intestinal hookworms. Both have the same life cycle. Their eggs also look the same under light microscopy. And so the main difference is going to be in the adults. Encyclostoma adults overall tend to be longer, where the males are 8 to 12 millimeters, and the females are about 10 to 15 millimeters. So something that you're going to see that is pretty much conserved through most nematodes is it tends to be the females that are longer instead of the males. So here is the life cycle of Encyclostoma and Necator. So first we just have development into a floweriform larva within the environment. Eventually this larva will then burrow through, penetrate the skin of a human. So after they penetrate the skin, they then circulate throughout the body. They eventually reach the lungs, and then when they're in the lungs, they can then be coughed up and swallowed. Then once they're swallowed, they then reach the intestine, and then the intestine is then where they actually colonize. So the adults then develop inside of the small intestine. They then release their eggs within the feces of the host. These larvae hatch in the environment, and then they develop in the environment, and then the cycle just repeats itself. So here are the eggs of these two parasites. They both look very similar to each other. They have very thin shells, and usually when they're in early mitosis, they'll contain 2 to 16 cells inside of them. So all of these little bundles you can see are the cells inside of the eggs. And then I have a video of what the larva inside an egg looks like. So here's the larvae of the two worms. This is the L1 larvae, meaning the most early stage of the larvae development. So it's a free-living organism, and it develops within soil or within the feces that's been excreted from the host. Usually they're not found in the feces. It's usually only if there's a delay in processing the sample that the eggs will begin to hatch. And so they're about 250 to 300 microns long. So here's the L3 form of the larvae, or the floweriform larvae, which is the infective form of the larvae. So they have a pointed tail or posterior end that you can see here in this bottom image. They use their teeth to burrow through the skin, and they can survive for about 3 to 4 weeks in the environment if the conditions are favorable. And they're about 500 to 700 microns long. So now moving on to the Encyclostoma adult, because the adults are really where we're going to see the differences between the two parasites. So like I mentioned earlier, typically for nematodes the male tends to be smaller than the female. So this one on the bottom is the male, the one on the top is the female. So the adults will reside within the intestines of the host. So both the male and the female have sharp teeth within their mouths. They both have their intestinal system. And then really what separates them is, of course, their reproductive system is different. And then the male has that copulatory bursa and spicule that uses to hold the female in place for reproduction or mating. So here is showing the sharp cutting teeth that Encyclostoma has in the black arrows, which is what they use just to eat any food that's inside of the intestines of the host. And then here is showing the Necator adult, and instead of having sharp cutting teeth, they kind of have what's referred to as cutting plates, so they're a little bit flatter than sharp teeth. And then so showing the copulatory bursa of the males, they both look different between Encyclostoma and Necator. And again, females don't have this structure. So in Encyclostoma, it's a little bit wider, or in Necator, it's a little bit narrower. So moving on to the next parasite is Encyclostoma caninium. So this is known as a zoonotic hookworm, and it's typically infecting cats and dogs. So it doesn't cause any intestinal disease in humans. What it instead causes in humans is known as cutaneous larva migrans, and what happens there is they only infect the skin. They don't go further than that, and they don't circulate around the body. They just stay in the skin of the human. And the eggs of this parasite are also indistinguishable from Ankylostoma duodenale and Necator americanus. So here's the life cycle of Ankylostoma caninium. So here we have the rhabitiform larvae within the environment. So they then penetrate the skin of a human, and in the skin of the human, that's it. They just stay in the skin, and they just cause these really ugly, long-looking lesions from them moving around in the skin. But then when you have a cat or a dog, when they penetrate the skin of a cat or a dog, they then migrate to the small intestine. And then once they colonize the small intestine, you then have the eggs being released from the feces of the animal definitive host. And then the larvae then hatch and develop within the environment, and the cycle just repeats itself. And for these definitive hosts, such as cats and dogs, it's possible for a pregnant mother to pass the parasite onto the developing puppies or kittens. So here's the Ankylostoma caninium adult. So it has very similar teeth to Ankylostoma duodenalis in that they're very sharp, and here it has 3 pairs of teeth, where Ankylostoma duodenale didn't have 3 pairs of teeth, it only had 1. So that's the main difference between their teeth. So moving on to the next parasite today is Ascaris lumbricoides. And this is the most common parasitic nematode in humans. It's also known as a giant roundworm, and it can reach up to 35 centimeters in length, making it also the largest parasitic nematode. So here's the life cycle of Ascaris. So this life cycle begins by ingesting the eggs instead of the parasite itself burrowing into you. So once you ingest the eggs then hatch within the intestines, and then the larvae, when they're in the intestines, they can leave and escape the intestines. They can enter the lungs, and then once they're coughed up, you then swallow them, and then you just keep perpetuating this cycle of you keep having larvae enter your intestines some more. And so eventually they mature into adults within the small intestine. The eggs are then released in the feces, and the egg at first is unfertilized when it first exits the feces, so it takes some time to become developed outside of the body. So once the egg is fertilized, then undergoes development. It then undergoes embryonation, which means that the cells inside the egg have now become a fully developed larvae. So when the eggs are passed in the feces, since they're not fertilized right away, they are not immediately infective. So it does take some time for them to become infective. And the female can actually produce up to 200,000 eggs per day. So that also allows for this infection to be very hard to get rid of, just because there's so many eggs being produced. So here's an unfertilized egg of Ascaris. It's overall a more elongated egg. It has a very thin shell, and they have what's called a mammillated layer. It's just a very bumpy perimeter, and overall this layer, the function's not really known, but you can see these kind of bumps and uneven surfaces out here, so that's what that layer is, and that's just how you can recognize an unfertilized egg. And then also inside the egg you can just see a bunch of cells inside that don't really have a very distinct shape. So now here's the fertilized egg. So the mammillated layer might still be there, it may not be there, and if it is there, it's not as bumpy as it looks in the unfertilized egg. And as you can see, it now looks more round, it's not as elongated, and now you can more clearly see the 2 cells that are eventually going to form the embryo. And so if they don't have a mammillated layer, it's referred to as a decorticated fertilized egg. So this one here, you don't see that mammillated layer. So overall, here's just a more clearer visual summary of the differences between the unfertilized and fertilized Ascaris eggs. So here, the unfertilized eggs, you overall just see that more elongated shape, and then here in the fertilized egg, more of a circular shape with a thinner mammillated layer. So moving on to the next parasite of today, Enterobius vermicularis. This is known as the human pinworm due to the female's long pointed tail. And so after mating, the male always dies, so that's unfortunate for the male. So the gravid female, meaning the female that's full of eggs, she'll emerge out of the host's rectum at night when the host is asleep in order to lay her eggs just outside of the host's body. And that's because the eggs require oxygen in order to mature. And after the female lays her eggs, she then dies too. So then here's the life cycle of Enterobius. So here we have embryonated eggs that would be ingested by a human. The larvae then hatched in the small intestine, and these worms will then just stay within the intestines. They don't circulate throughout the body like how the previous worms we talked about do. So the adults will hang out within the large intestine, and then a gravid female will then actually just escape and leave the colon, come out of the host rectum. She'll then lay her eggs on the perineal folds of the host, and then these eggs will then mature within 4 to 6 hours. So here's an image of the eggs of Enterobius. So they're very elongate to oval shape, and so a way to diagnose these eggs is they can actually stick to tape. So you can use like tape to just collect the eggs from a host, and then also the eggs fluoresce under UV light. So here's showing an image of how the eggs fluoresce. And then here is just an image of unstained eggs just under light microscopy. So here's a comparison between the Enterobius male at the top and the female at the bottom. And so here you can see the female's very long, sharp tail, which again gives it the name the pinworm. And the males have a more blunt end with a single spicule. And both of them have cephalic expansions, meaning that their head just looks a little bit narrower and longer. And so here's a close-up view of the posterior end of the male, being almost flat, blunt looking, and then the female's pointy posterior end. So moving on to the next parasite of today, we have Strongyloides cercoralis. And in this parasite, only the female is parasitic. So since only the female is parasitic in this worm, only the female will be found within the intestines of the human, and so the eggs will be produced by parthenogenesis by the female, and so the offspring will be exact clones of the mother, since they will have her exact same genetic information. And these are referred to as the threadworms. And so for this parasite, the eggs hatch within the small intestine instead of out in the environment, so it's very rare to find any larvae within the feces of the host. So here's the life cycle of Strongyloides. So this life cycle is kind of complex in that there's a free living cycle and then there's a parasitic cycle. So in the free living cycle, what happens is you have free living adults just developing within the soil, and then the adults will then produce fertilized eggs. The eggs then hatch to form larvae. The larvae will then eventually develop into the Phylariform larvae, or the L3 larvae, and then this larvae will then penetrate the skin of a human host. And so once they're within the human host, they'll then migrate by various pathways to the small intestine, and then once they're in the small intestine, they'll then develop into an adult. And so once these larvae enter the intestine, they mature into adults. Once the larvae enter the human body, they'll never mature into males, they only mature into females. And exactly why is this, it's not known, but there are no parasitic males, so you'll never find any males within the small intestine of a human host. Something else that the Phylariform larvae can do is that it can also escape the intestine, and it can also eventually circulate out into the lungs. And then you can have auto-infection, which is when you can then cough up these parasites and then swallow them, and then they'll again just enter your small intestine again. So the females then produce eggs through parthenogenesis, where they're not fertilized by a male, but instead they just contain copies of the female's genetic material, so these eggs will be clones of the female. So the eggs will be deposited in the intestinal mucosa. Eventually the larvae will then hatch, within the intestines, and then they'll then migrate to the intestinal lumen, and then these rabidiform larvae then hatch within the intestine, and then they're excreted in the stool. And then they can again either go on to infect another human, or they develop into the adults and live a free lifestyle in which they can then reproduce in a typical manner by having a male fertilize a female. So here's the male and female adult form of Strongyloides. So both of these are the free-living form of the parasite. Here's the free-living male on the left, and then the free-living female on the right. And all of these circles that you can see within the female, that is a row of eggs that's visible on the inside, and then the male has this very sharp spicule on the posterior end. And so here's what the parasitic female looks like. This would be the female that's inside of the human host. So she's much more elongated than the form when she's free living outside of the body. And this would be a transverse or cross section of the female. You can see her ovary. Here you can see an egg inside of her body, and then you can see her intestines. So here's the rabidiform larvae of Strongyloides. So here we can see the buccal canal, their mouth. You can see the median bulb of the esophagus, which is then just going to eventually branch into the rest of the esophagus. Here's a more zoomed out picture that just shows again the mouth, the median bulb of the esophagus. Here's a genital primordium, which will further develop to either become a male or a female genital area. And then something that's unique about the Strongyloides larvae is that the tail is forked at the end. So moving on to the next parasite of today, we have Trichuris. So Trichuris is known as the human whipworm, and only individuals with a heavy parasitic burden will become symptomatic. So only individuals who ingest a very large amount of parasites will become symptomatic. And so it takes about 3 months after the ingestion of eggs for an individual to actually start showing symptoms, because that's just how long it takes for the mature worm to actually develop. And the females can grow up to 50 millimeters in length. So overall, this is a pretty problematic parasite since it can go undiagnosed for such a long time. So here's a life cycle of Trichuris. So the way a human gets infected would be by ingesting embryonated eggs through the contaminated environments. The larvae will then reach the small intestine, and then they'll hatch in the small intestine. The adults will then colonize the cecum of the large intestine. The adults will then fertilize each other, and then they'll produce unembryonated eggs. These eggs are then released in the feces, and in the environment, the eggs then become embryonated, and they develop further in order to have the little larvae inside the egg. So here's what the Trichuris egg looks like. So they're referred to as barrel-shaped, and they have these polar plugs at the opposite ends of the eggs. They look very thickened in shape, so that's something that's unique to this parasite. And like I mentioned earlier, the eggs are unembryonated when they're passed in the stools, and they're about 50 to 55 microns in length. And sometimes you can have some eggs that are a little bit atypical in their shape, in a sense that this one is kind of more rounded than it is elongated, or the polar plugs can be a little bit uneven, like how this one is flatter and this one is wider, and how, for example, on this bottom egg, you'll see on the left side you can see the polar plug pretty clearly, but on the right side it's more difficult to see. So overall you can see atypical eggs that don't show the very typical features of the parasite very clearly. So moving on to the Trichuris adult. So males have a coiled posterior. It can be seen clearly in this right image. You can see that the posterior end goes into like a spiral shape. So that's what it means that it's coiled. And then you can see the spicule also on the end of their tails. It's a little bit visible here in this image too. And then the females instead will have a straight posterior, so it won't look all spiral like this. And both worms have a whip-like anterior end, the way it's very elongated than the rest of the worm. So both males and females have that worm, and that's what gives them the name whipworm. So here's a more clear image of the male. Here's their coiled posterior end, and then the spicule is very clear in this image. And then also that very elongated and wavy whip-like anterior end is also very prominent in this image. So moving on to our next parasite, it's Diofilaria. So this is known as the heartworm that infects dogs. So dogs are known as the definitive host of this parasite, but it can also infect other mammals such as wolves, cats, ferrets, bears, and humans. However, these infections are very rare, so it's much more likely to impact dogs, which is usually why typically dogs are vaccinated for heartworm. And so dogs who live a sedentary lifestyle, or dogs that tend to just lay around all day, those dogs may not show the symptoms if they have the disease. And if a human does get this disease, they may have symptoms such as pulmonary granulomas or subcutaneous infections, so they can actually form lesions within their lungs or infections underneath their skin. So here is the life cycle of Diofilaria. So the way both a dog and a human would get infected by this parasite is the same method. It has to take a mosquito that is infected with this parasite in order to bite either the dog or the human, and then the L3 larvae, or the filariform larvae, will then enter the bloodstream of the human or the dog. And so what can happen in the human is they can eventually travel to the lungs within the humans, and in a severe infection in humans, they can eventually form very small coin lesions that are visible within the lungs on radiographs. And then when they're within the dog, they instead will travel to the heart instead of the lungs. And they'll be within the pulmonary arteries of the heart. And if you've ever seen, I don't know, you could actually look up images of heartworms in dogs, and you'll actually see images where the heart is actually just bursting full of worms. It's pretty dramatic for a dog to have severe heartworm infection. So when they're in the heart, the adults will then produce another life cycle stage known as the microfilariae, and those will then be found within the peripheral blood, and that's the blood that just travels throughout the veins that eventually will travel through the heart and other arteries or capillaries. And so the worms can actually survive for 5 to 10 years within the heart of the dog. So as the microfilariae are circulating around the blood, a mosquito has to then eventually bite the dog again, and then the mosquito will then ingest these microfilariae. And so the microfilariae will then penetrate the mosquito's midgut, and they'll migrate to the mosquito's abdomen. The microfilariae will then develop into L1 larvae, and eventually they'll mature into L3 larvae, and then they then migrate to the mosquito's proboscis, which is their mouth, and then they'll then be ready to be injected again into another host. So here's an image of the adults of Diofilaria imidis. So they're just very string-like in shape. So the female is longer, and the female can be about 10 to 12 inches in length, where the male will only be 4 to 6 inches in length, and the male will always have a very coiled, curly-looking tail. And so here's an image of the microfilariae that circulate through the bloodstream, and they can circulate for as long as 2 years in the bloodstream, and eventually they'll be ingested by a mosquito. So moving on to the next parasite of today, it's Wucheria bancrofti. So this parasite is the common causative agent of lymphatic filariasis, and it eventually causes elephantitis, which is enlargement of certain limbs. And this parasite is similar to Diofilaria, but it only infects humans. And so worms mature in about 6 to 8 months. The male and the female worms mate, and then they'll release the microfilariae, and then the microfilariae can actually continue to be released for up to 10 years within the human host. So here's a life cycle of Wucheria. So it starts when a mosquito bites a human, and then the L3 larvae will then enter the skin of the human. And so what happens is they migrate to the lymphatic system, actually, and then they mature into adults within the lymphatic system. So the adults then produce sheathed microfilariae, meaning that they just have a little protective cuticle around them. And then they then migrate throughout the lymphatic system, and then they eventually reach the peripheral blood circulation. So they'll eventually start to circulate throughout the veins of the body. So then what will happen next is a mosquito will then take another blood meal, and it will ingest these microfilariae. And once the microfilariae are inside the mosquito, they'll then lose their sheaths. They then penetrate the mosquito's midgut, and they migrate to the thoracic muscles of the mosquito, and then they're able to develop further into the L1 larvae and then the L3 larvae. Then eventually they head to the mosquito's proboscis, and then the cycle then just repeats itself. So here the upper image shows a cross-section of an adult female within a lymph node. So A is showing the intestine, B is showing the uterine tubes of a female, C would be the lateral cords that can help the parasite move, and D is just a thick cuticle that forms over these cords that can help form the protective sheath that it has when it's circulating through the human blood. The lateral cords also enclose the excretory canal. And here the bottom image shows a male worm on the left and a female worm on the right. So moving on to the next parasite, called the Trinchinella spiralis. This is the causative agent of trinchinellosis, and this disease actually infects a wide variety of mammals, and it also has a sylvatic cycle, which means that it only infects wild animals in this particular cycle. So adult Trinchinella species reside within the intestinal tract of the vertebrate host, and then the larvae can then be found encapsulated in muscle tissue. So here's the life cycle of Trinchinella. So like I mentioned, there's the sylvatic cycle that infects wild animals, and then there's a domestic cycle that tends to infect like cattle animals. So in these cattle animals, you'll have the encysted larvae within the striated muscle. And so the way a human would get infected would be through ingesting undercooked meat through one of these cattle-like animals. A human can also get infected from an animal from the sylvatic cycle, also by hunting maybe one of these wild animals and then ingesting the undercooked meat from one of those animals. And these animals will also have the larvae encysted within their muscles. And so the way these sylvatic animals get infected is through predation and scavenging other smaller mammals, where the way the domestic animals will get infected is through eating small meat scraps or even cannibalism. And so once inside the human, this parasite will then release its larvae in the small intestine. The adults will then reproduce within the small intestine, and then the larvae will eventually be deposited in the mucosa of the intestinal wall. Eventually these larvae can then escape and enter the circulation, and then that's when they'll reach the muscle, and then they'll encyst into a little cyst form and just stay there for many years. So here's a diagram of the Trinchinella adult. And so the male and the female overall look similar, but again, the most obvious way to tell them apart would be the male's copulatory bursa. And so here's what the encysted larvae within the muscle look like. And so when the larvae have been in the muscles and inside their cyst form long enough, they'll become called stichocyte. And these just contain secretory granules that can impact the area around the cyst. So here we see in LC the lateral cords, IN would be the intestine of the larvae. And then RT would be the developing reproductive tubes. And then to the right, we just see more detailed images of these cysts within the muscle cells. And the way their name is Trichinella spiralis, so you can see that they give a clear spiral-like shape when they're in their cyst form, so that helps give them their name. And so here's a more clear image of the larvae of Trichinella. So they also coil into a little spiral, which also gives them their name when they're in their excysted form. So moving on to the last parasite of today, it's called Onchocerca volvulus, and this causes the disease known as river blindness, and it's the second leading infectious cause of blindness throughout the world, and the average lifespan of the adult worm is about 15 years. So this disease tends to begin with severe itching, and long-term infection can result in keratitis, which is inflammation of the cornea, and then eventually, if untreated, it can lead to blindness. And Similium, also known as the black fly, is the vector for O. volvulus. So here is the life cycle of O. volvulus. So it begins when the black fly, or Similium, takes a blood meal, and through the bite wound, the L3 larvae will then enter the wound. So they then burrow deep through the skin, and they reach the subcutaneous tissues. Adults will then stay in the subcutaneous tissue within a subcutaneous nodule. The adults then produce unsheathed microfilariae, and these microfilariae can be found within the skin, and they can eventually reach the lymphatics and connective tissues. They can also reach the blood and the urine and the sputum, which the sputum is phlegm that you cough up in your lungs. They can reach these places by entering the bloodstream, by burrowing through capillaries. So another black fly will then come and take another blood meal, and it will ingest these microfilariae. The microfilariae will then penetrate the black fly's midgut, and then they'll reach the thoracic muscles. They'll then develop into the L1 larvae, eventually developing into the L3 larvae, and then they'll then migrate to the proboscis of the black fly, and then the cycle will then be able to repeat itself. So here is a cross-section of an adult female worm within a human. So here we can see the uterine tubes that will allow the eggs to pass through. Here's the intestine, and in the green arrows are the cuticular nodules, and then the right image shows an even more magnified image of an adult worm, and here you can actually see the microfilariae developing within the worm's uterus. So here's a visual summary of the various posterior ends and the various copulatory bursas of the different nematodes that we talked about today. So overall, these posterior ends and copulatory bursas are probably one of the best identifying features of the adult worms, and also the best way to differentiate the males from the females. So here's the one that we just finished talking about, Onchocerca. So it has its spicule. There's Encyclostoma, female worm, just has a very pointed posterior end. We talked about Ascaris, and then an Encyclostoma, male worm, has a very wide copulatory bursa. And with that, I hope you learned something new, and I hope you have a good day.